Today we're going to be talking about meiosis, and it is the division of sex cells. In the last lecture when we talked about mitosis, we learned that the cell will go through a process of cell division that allows the one cell form into two new daughter cells that are exactly the same as the original cell. Now the big question that many people come up with, well, how do offspring come about then? If this is the process of cell division. If this were to take place, then if a human has 46 chromosomes, that means its offspring would have twice as many when you join the male and female reproductive cells together. Well, there's a special process called meiosis that allows us to cut this number of chromosomes in half. And today we're going to be taking a look at that. So what is meiosis? Meiosis is a process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Now, some of that may sound confusing. Think of it this way. The diploid cell is just your normal cell that's going to be split into two, and the diploid cell has two sets of chromosomes. You have 23 sets of chromosomes in your body. You have 23 from your mother, 23 from your father. Now, the homologous pair basically means that both of these are going to carry the same genetic information. And then when we get talking about genetics, then we start seeing how dominant and recessive traits come from the pairing of these homologous pairs. Now, a haploid is one set of chromosomes, and that means a haploid cell is only going to have one set. It's not going to be paired. So essentially, what we are doing in a human, you have 23 pairs. We are going to figure out a way to split those up to where there's no longer any pairs. Now, what does homologous mean? Chromosomes that each have a corresponding chromosome from the opposite sex parent. And that's what I basically mentioned before. You have 23 from your mother, 23 from your father, and these two will line up, and each pair will carry the same genetic information like hair color, eye color. Any of these things are going to be included on your homologous pairs. Now, let's take a look at what the pairs are actually. Here are the 23 that we mentioned before in a mapping, and then you can see each one is paired up. Let's say the left is from your father and the right is from your mother, or actually it's vice versa, that's flipped. But when we take a look at that, you're going to see, okay, these are how this all matches up and how they work together. But let's take it a little further. What does it mean, homologous pairs? How does that work? Well, in a cell, you're going to notice that both the chromatids have made copies, but something happens in meiosis that is very unique and allows for variety. And what that is as the process changes, we have these homologous pairs, and they start to switch, and they change, and they take parts from others, and that allows for variety. And we'll talk more about this as we get going into meiosis. And again, this is a little more advanced level, but it's something that we are going to be just discussing very briefly. Meiosis stages. Meiosis usually involves two distinct stages, meiosis 1 and meiosis 2. We're going to first start with meiosis 1. Now, today what I've got is a short little video that just kind of shows what's happening in the cell. And you notice this is very similar to as before. The chromatin starts to wind. As it starts to wind up, we see the chromatids, they are copied, and they are right next to each other. Now, this is where the variety comes into play, where you'll notice where they will switch and change out different alleles, different little genetic code, and this is what creates variety. This is why you're not the same as your brother or sister. and you'll see the transition here. Now again, just like in mitosis, the chromosomes will go to the center. Now, but here's the difference. In metaphase 1 of meiosis, they don't line up on the equator, they line up at. Notice the pairs are separated. They're not actually going to split the pairs up. The pairs are going to go to separate sides. So now we have two new cells in telophase 1. We have two new cells that are exactly different. So we have 3 original chromosomes in the left, and we have 3 original chromosomes on the right, and they're totally different cells. And notice what happens again. Spindles go to the corners. Now this time in Metaphase II, we see them actually split the pairs apart and form 4 haploid cells. And if we take a look at this, there's no pairs in here. We have 3 chromosomes in each when we started with a cell that had 6. And that's what the haploid cells are. So let's talk about these. Prophase 1. Each chromosome pairs with its corresponding homologous chromosome, form a tetrad. Again, that's not a term that we're going to use, but later on in Mr. Shidley's class or down the road, you will hear the word tetrad. There are 4 chromosomes. The pairing of homologous chromosomes is key to understanding meiosis. So when they pair up, you got to understand that these chromosomes will pair. They'll be right there next to each other. And if you can get that down, like for a human, for instance, there's 23 pairs. So all the hair color, eye color, all that information from your mom, it's going to pair up with the same chromosome from your father. Crossing over may occur here. So again, that's what I talked about before, the crossing over of genetic codes and switching and creating a new look or whatever towards the genetic code. That's possible during Prophase 1. Prophase 1, let's take a look at what it looks like. Obviously the chromatids, it looks exactly like prophase of mitosis. The chromatin has unwound to form chromatids. The chromatids have copied. They have exact replicas of each other. And then you have the site, the chiasma, the site of the crossing over, where they can cross over if they choose to do so. And this is the point right here in prophase where there's just that little addition. Notice we'll also see that the nuclear membrane starts to disappear. Then we move to metaphase 1. Now metaphase 1, the spindle fiber is attached to the chromosomes. They line up at the equator. Notice it's not on the equator now. We don't put that centromere, we don't put that bridge right on the equator because we're not going to split the pairs up. We are going to take each pair and pull 2 pair to each side. And now we'll have 2 totally different cells when we make this first switch. So that moves us to Anaphase 1. And again, moving fairly quickly through here, just so you guys can, keep this a shorter type of lecture, and that way you learn a little bit more as you keep going through this. Anaphase 1, the fibers pull the homologous chromosomes towards the opposite ends. So really you see that in these phases, they're all doing the same thing. There's just a few key changes in here. So you have Anaphase 1 where the fibers are pull that homologous pair across. And we can see here, if we take a look over here, we have 2 different pair. So we have a total of 1, 2, 3, 4 chromosomes, but only 2 original. Then over here, 1, 2, 3, 4, and only 2 original. And these are not the same as these over here. So that's going to give us an opportunity to have variety. That leads us to telophase 1 and cytokinesis. The nuclear membrane forms back. The cell separates into 2 cells. And it actually does separate into 2 cells. So we see this when we separate into 2 new cells. And you'll notice we have 2 new cells. Then we go back through, again, go to Prophase 2, where now we're starting Meiosis 2. Meiosis 1 results in 2 haploid cells, and we notice that there's two cells there. Each cell has half the number of chromosomes as the original cell. So we'll notice that now in each cell we have only 2 original, instead we had 4. Now here's Prophase II. Again, what's happening at Prophase II? Really, the spindles are going to the ends. The centrioles are going to the ends to start the fibers to attach. Nuclear membrane starts to dissolve once again. Leads us to Metaphase II. This is where the chromosomes line up, similar to metaphase in mitosis. So they line up on the equator. So notice what's happening here. We have it line up on the equator. So that bridge is right here. We're lining up on that equator, and we're getting ready to split these cells. That leads us to Anaphase II. And again, what does Anaphase do? Anaphase is where they separate and move to opposite ends of the cells. It pulls those the chromatids apart. And this is kind of a comparison. In Anaphase I, it pulls the pairs. We have the 2 pairs. And then Anaphase II, we're splitting the pairs up. Notice we have 1, 2, 3, 4, 1, 2, 3, 4. We have all new variety. Now, and we would have only this cell over here in Anaphase II would look exactly like this side too, except we had that crossover gene that switched it up, that we had a crossover that allowed it to have variety, have a little bit of difference out of this side over here, if you can see where they touched and they cross over. Then we have Telophase II. This is where Meiosis II results in the 4 haploid cells. And what do we see in here? We see the nuclear membrane start to come back, and we see 4 new cells. And what are these cells? These cells are sex cells. In the males, meiosis results in sperm. In females, meiosis results in egg cells. And these cells are produced to allow us as humans, as animals, as plants, to reproduce sexually. And it allows the sperm and egg to join. Sperm has 23 chromosomes for the human. Egg has 23 chromosomes, and those 2 will join together and form 46 in one cell. And what happens right after the forming of the 2? Then we see mitosis start to take. So we take a look at mitosis versus meiosis and kind of see what the difference are. In mitosis, again, the results are 2 diploid cells, which in a way they label it 2N, and I just call it the same. They're the exact same. So they're sister cells. So in mitosis, what's the result? We have the same cells. So as the original. We have 2 of them, but they're the exact same. Then we take a look at meiosis. What do we have as a result in meiosis? We have 4 haploid cells, 1N, and I look at it as we have 4 new cells that are half. They have half the number of chromosomes that the original had, and that allows us to have sexual reproduction. Then we have, in mitosis, cells are genetically identical. They're exactly the same in mitosis. They don't sway, they don't cross over, they don't switch. They're exactly the same. And then in meiosis, they are genetically different. This is what brings about variety. In mitosis, we also see that they form the somatic, the body cells, and so it forms your body, your bones, your blood, your skin, your hair. All the body cells are formed through mitosis, and in meiosis, it forms the sex cells, the sperm and the egg. Now, one big difference between the 2, and you take a look at the process altogether, it's found in metaphase. Metaphase I and metaphase of mitosis. If you take a look at this and you know, and you've been paying attention, you know which one is metaphase of mitosis and which one's metaphase of meiosis. The one on the left is metaphase I of meiosis, and the one on the right is metaphase of mitosis. They line up on the equator in metaphase, splitting the 2 new cells, 4 and 4. And off to the left, we see the splitting of the 2 pairs, but they don't line up on, they line up at the equator. Notice their centromere, the middle, is not on the equator, and it's going to split these this way, to the left and to the right, giving us 2 and 2, which are 2½ This is meiosis, and this is what allows cells to form the sex cells. So we have the reproduction possibility through sexual reproduction, and from this is how we have the two processes that allow life. And I hope that you guys continue to study and just keep getting ready as we get ready for genetics.